So salmonella, probably I should rename my slide paratyphoid salmonella after hearing to your talk. The biggest problem with salmonella is asymptomatic, meaning, for example, the lethal dose for a mice is 100 colony forming units. But in chicken, we put 1,000 million, 1 to the 10 to the power of 9 in a grown adult chicken. They don't care, Back where I come from, India, salmonella is not a problem because we basically nuke the meat. We cook it so much. But here, people don't cook the meat. If you are like my wife, the first person she wants to complain about anything is me. So first she would complain me if she doesn't cook good. And then she wants to cook the chicken producers. Like that. They have to produce salmonella-free meat. So here we are, salmonella is pretty much asymptomatic, and it can cause disease in humans, and because of that, we always try to find ways to reduce salmonella colonization in chickens. Probiotics is one of the approach taken by the industry for. When you are talking about control of salmonella, there is no one step. For example, if you want to control smallpox, rabies, one step, vaccine, you're done. But with salmonella, you don't have a one-step process to control salmonella. So you do tons of stuff. One of the approach taken by the industry and has been proven successful to a big extent is probiotics. Probiotics are live microbial supplement, mostly supplemented as spores, even though we call it live, because most of the live microbes are not going to survive the gastric acid. And they have tons of immune stimulating properties, competitive exclusion of pathogenic microbes. They produce antimicrobial substance. And the product we worked in the lab is Poultry ME, which several of you call probiotic, and apparently it turns out it's a Synbiotic, which I will come to in a minute. Lactobacillus, it has 4 strains, Lactobacillus, Enterococcus, Bifidobacterium, and Pediococcus. So it's symbiotic, call it symbiotic. Symbiotic is a combination of probiotics and prebiotics. Prebiotics is the word coined by Robert Freud in 1995. Obviously, it's not here because I'm good looking than this guy, Robert Freud. So, in a nutshell, prebiotics are like food for probiotics. They are carbohydrates, and they are normally cannot be fermented by every microorganism in the gut. Some of these bacteria, these good bacteria, particularly bifidobacteria, they have this enzyme beta fructosidase 1,3 fructosidase, which can utilize these prebiotics. The product I worked with, Poultry ME, as fructose oligosaccharide, and with the hope that this product, fructose oligosaccharide, can grow the bifidobacteria. Preferentially, and this would help you to competitively exclude salmonella. So we did a couple of experiments. We have a system in the lab where we can basically screen thousands of individual bacteria for their ability to suppress salmonella or E. coli. We have a system in the lab. We do 2 things. One is as the effect of the supernatant in their ability to inhibit salmonella, or we can do a solid media, we can plate it in the media. I'm going to show the data with just the liquid media. It's pretty simple, straightforward. Once you set it up, it is much easier. You can screen lot of bacteria. The only problem is you need to have the individual strain, which, after a lot of cajoling, Raj was kind enough to send it to me. Actually, I had to send him some pictures, like beautiful high definition pictures of his dream girl, which turns out to be Angelina Jolie. So after a lot of those pictures, he did send me those individual strain with all those individual strains. So what we do here is take Salmonella, grow them inside, first grow them for 12 hours and so that we know the count of Salmonella, how much we have, so that it will be uniform across all the treatment. And then we take the individual probiotic strains and then we grow them. Either we do both things. One, we take just the supernatant and then we do a serial dilution of supernatant. For the supernatant is to Salmonella ratio. We try different ratio. We do it 3 times. The other option is you can just co-culture the probiotic strain with Salmonella and then plate it in the plate. So we did this second one I just talked about. And of all the species, here is the control group where the Salmonella is allowed to grow without any of the supernatant. So they do grow. And if you look, here is the 1:10 dilution of one of their Enterococcus, one of their species in their probiotic combination, Enterococcus, Lactobacillus and Bifidobacteria. Most of them are pretty efficient at 10:1 dilution, but as you dilute the supernatant, they were losing their inhibitory ability against Salmonella. So we got this data and it was pretty good that we wanted to move forward with in vitro model towards the in vivo model to show whether applying Salmonella applying these probiotics will help you in decreasing the Salmonella load in an experimental Salmonella infection. The second study we did was in vivo. We had a couple of objectives. The obvious objective was whether this Synbiotic product can decrease Salmonella clearance following an experimental challenge. As this is an industry project, we also looked at the performance, bird performance following the Synbiotic supplementation. Synbiotic is the word, it's a technical word where we use to, when a product has both a probiotic and a prebiotic, and it turns out Poultry Star does have both the probiotic and the prebiotics fructose oligosaccharide. So experimental design, it will change a little bit based on what was the age of the bird. Until age day 14, week 14, it was an extremely simple step, simple experimental design, a control group and Synbiotic, each of them at 24 cages and 4 species of probiotics and the fructose oligosaccharide, making it into a Synbiotic, 8 chicks per replication, 24 cages. At 14 weeks of age, we went for vaccination of the 24 birds in each of the group. 16 of them were vaccinated, 8 of them did not get vaccination. So until until we did the Salmonella challenge. So whatever you see before Salmonella challenge, it's basically a 2x2 factorial design with control and Synbiotic, vaccinated and unvaccinated are the main effects. And then at 23 weeks of age, birds were challenged. Half of the birds in this group with the vaccinated group were challenged with Salmonella and the rest were not challenged. So eventually it was, I hope we had a no challenge group in this group too, but this experimental design came from the God, even though he's not good looking. I had to follow the word of the God, so which we did. And so we ended up having a 3x2 factorial design with 2 levels of Synbiotic, and these 3 were the 3 different group. We were feeding 1 kg per ton of feed for the first 3 weeks, reduced the dose by half until between week 3 and 23. At 23 week of age, when we did the Salmonella challenge, we increased the dose back to a kg per ton of feed. The vaccination we did was with Poulvac. It's subcutaneously killed vaccine at 2 ages, week 14 and week 17 is the booster dose. Challenged with a huge dose, 1 to the 10 to the power of 9 CFU of Salmonella enteritis orally on week 24. We measured body weight, though we don't measure body weight in layer birds. We measured because we were seeing the birds that were fed the Synbiotic were coming into production early. It was, my graduate student came and said that, hey, these birds that were fed Synbiotic are coming up in production very early. They are coming up pretty fast. So we measured body weight because of that reason. And then we pulled out birds at each time point. To measure some of the immunological measurement. So this is the body weight, like 2 by 2 factorial. So body weight was significant. This is the group that got the Synbiotic. Surprisingly, vaccine had some effect. Because of that, I have to maintain 4 groups. Otherwise, I would have pooled some of the main effect. So it turns out birds that were fed Synbiotic were gaining body weight much faster, like compared to the group that were not having the Synbiotic. There was little bit effect of vaccine on the birds that were fed Synbiotic, that it did decrease body weight a little bit. The Salmonella killed vaccine. We were seeing this body weight until 20 weeks of age. After that, it disappeared. The birds that were not fed Synbiotic caught up with the birds that were fed Synbiotic at 20 weeks of age. So eventually, this, I think, led to the increase, like once the increase in body weight basically translated into the birds coming into egg production much earlier. The birds that were, like I pulled out the effect of vaccine at this stage because the effect of vaccine was not significant. So I'm presenting the main effect of only the Synbiotic. Birds that were fed the Synbiotic came into production, egg production much earlier than the birds that were not fed Synbiotic. And this we were seeing even until 23 weeks of age. So at this point, we did challenge the birds with Salmonella, and even after Salmonella challenge, birds that were fed Synbiotic were having a high production of, had an increased amount of hen day egg production even at after Salmonella infection. Most of the weeks, even some of the weeks, they were not statistically significant, but eventually there was some statistical significance. But as you would see, like the end-to-end production were going down after a while, not because these birds were not in production. We were starting to kill birds. We were collecting samples, so we were losing some birds. Because of that, the end-day egg production started to get a little bit messed up after some point. The other thing I always ask, like people is, how do we know that the probiotic products we are feeding even survive in the gut? It's the example I give is it's like an Amazon jungle. Doesn't matter how much Raj likes Angelina Jolie. If I'm going to leave Raj and Angelina Jolie in Amazon jungle and go after 30 years with the hope that they would have repopulated the Amazon jungle, good luck. It's not going to happen. It's like a jungle out there. So when you try to try to put your probiotics product, how do we know that they even survive the gut? So I try to, it is an extremely complicated step to do because we have tons of microorganisms. Even the same species, Lactobacillus has several species. So it was a tough thing to do, but we came up with some of primers that were unique to this product Biomin was having. And we were showing that we were not able to identify every probiotic, but at least we were able to find a couple of, or probably say of the 4, we were able to identify 3 of them. So what we were showing is, basically, if I'm going to ignore, there are some variation here. if I'm going to look at the effect of symbiotic, there was a pretty much effect of symbiotic on this on the existence, on the survival of Lactobacillus acidolacti in the gut. We were able to identify this species at a relatively low proportion, but still we were able to see that the control birds did not have this particular strain of Pediococcus, but the supplemented group did have that. Same with Lactobacillus reuteri, we were able to identify this particular strain in birds that were challenged with Salmonella. Surprisingly, the group that were challenged with Salmonella had a high amount of this. So in this figure, the last three groups are the groups that were supplemented with symbiotic. The rest were not supplemented with symbiotic. Sometimes we do see birds that were not supplemented with symbiotic having this particular species strain of Lactobacillus. Surprisingly, we don't know how it happened, but the group that were supplemented had a high amount of Lactobacillus reuteri, and this number went up in the groups that had Salmonella challenge. This is the most important thing, the Salmonella load in the Zika. What we found was birds that were vaccinated but had no challenge. Obviously, they did not have any Salmonella. But the birds that were challenged, no vaccine, they had the highest load of Salmonella in the Zika. Birds that were vaccinated and didn't challenged, the Salmonella load came down. Interestingly, the birds that were fed symbiotic and challenged were really much as good as the bird that were vaccinated with Salmonella, but they didn't, like the birds that were fed just a symbiotic, this group was really good compared to the group that was fed vaccine. A couple more minutes left. So we also measured at IgA, Salmonella IgA. What we found was the birds that were challenged with Salmonella had a high amount of IgA, which was surprising because we were hoping that symbiotics groups would have a lot of IgA. And then what we think is happening is this group had this is the primary immune response, because these birds were never ever exposed to Salmonella, and because of that, the primary immune response tends to have more IgA compared to the secondary one, where we tend to have less antibody but more affinity. I think that's what is happening here. We looked at interleukin 10. Interleukin 10 is an anti-inflammatory cytokine, and some of the symbiotics, I work with a lot of them, are notoriously known for upregulating interleukin 10. And we found that this product does seem to upregulate interleukin 10, which could be bad because we need inflammation to fight the infection. Like for example, in the birds that have Salmonella, you will see low amount of interleukin 10. We were surprised that this symbiotic product did increase interleukin 10, but it turns out they also upregulate inflammatory cytokine, tumor necrosis factor alpha, which you need to fight infection, to fight the intracellular pathogen like Salmonella. We looked at CD4 cells. I'm going to just skip because I have only 22 seconds. Basically, all these groups were comparable. Once you have a Salmonella infection, we'll have a lot of CD4 cells, CD8 cells, but all the groups are pretty much comparable in CD8, CD4 too, CD4. We looked at T regulatory cells too. To conclude is, what we felt was when you supplement symbiotic product, it's beneficial in increasing layer production performance and protection against Salmonella infection. Thanks to my lab people, the farm crew. They are my mentors, Geeta, Kirk, my master's boss, my PhD boss, and postdoc boss. They are the people who made me who I am today. So wherever you guys are, thank you. Thanks, Biomin. My wife apparently we speakers get some honorarium. My wife wants to thank Biomin for the honorarium because she did spend the money already. So thank you, and with that I'll take your questions.
Thank you. Thank you, Associate Professor Ramesh. Can I invite you to ask questions? Any questions? No? Then it's my honor to ask the first question. Ramesh, I'm very interested in how good bacteria, probiotics, actually can stimulate immunity in terms of communication with the body to produce immune cells in an optimized way.
So it's not just, like good bacteria. You put any bacteria in the gut, they will stimulate the immune response. This work have been done with germ-free mice. Mice that have never been exposed to germs in the gut. Their gut immune system is totally undeveloped. So basically, you put any bacteria, they will stimulate the immune response because doesn't matter whether it's a good bacteria or bad bacteria, they are foreign to our immune system. So they do develop immune system. But the good thing with the probiotic is they don't cause damage. So basically, we have M cells in our gut where they, they can go get these antigens from the bacteria. They have some of the pathogen-associated molecular patterns. For example, their glycosylation pattern of the bacteria is different from our glycosylation pattern. So basically, our immune system can recognize these patterns and stimulate the immunity without the accompanying damage by the bad bacteria. So in a way, good bacteria helps us to stimulate our immune system.
So it's a cell-mediated immunity.
That's right, cell-mediated immunity.
Thank you. Do we have any other questions for Ramesh?
Thank you.
I actually have one more question before I let you off. I'm a very inquisitive person. I'm just wondering why pathogenic bacteria cannot actually utilize prebiotics like fructooligosaccharides. Why is it limited to bifidobacterium and
great
lactobacilli?
question. So most of the prebiotics are oligosaccharide. Basically, they are glucose unit and fructose unit. But the way we link them has a particular linkage. And that linkage can be broken down by an enzyme beta-1,3 fructosidase. And only some of the bacteria have this enzyme to break down this bond. So once they break down, they basically have glucose and fructose, which they can use as an energy. But if they can't break down, it's a polymer which cannot be fermented by those bacteria. So this gives a competitive advantage for bifidobacteria and some of the beneficial microorganisms.
Thank you very much.
Okay.
Thank you, Ramesh.